Okay, so in today's video we will analyze a little bit RNASeq data. So there will be a few videos on RNASeq, which means that we will use NGS to study the gene expression levels. So first I will clarify a little bit the terminology. By gene expression, in fact, we mean gene transcription. After maturation. Which means that we count, or we try to count a little bit, how much mature mRNA molecules exist. So usually, when we study gene expression, we study the levels of mRNA. Not of all RNA, actually, but only of mRNA. And we achieve that by sequencing. This is a breakthrough of the NGS technology that can achieve different things, like gene expression or study of polymorphisms or study of binding of transcription factors, by a common thing, which is sequencing. Okay, so let's see now the principles of this and how it works. So let's imagine that we have a population of cells. So we have a tissue here. And of course it's composed by some cells. Okay, and in this cell, this is the genome, for example, and the genes are over here. I will just use another color for the genes. So I have a blue gene and a blue gene and a red gene. And what we do is that, and for example, the red gene produces a lot of transcripts. So this is mRNA now. And the blue gene produces a few transcripts. And we assume that this is what's happening more or less in all the cells of this tissue. So we have the DNA here. Again, we have the red gene, and we have the blue gene, and a few transcripts from the blue, and several transcripts from the red. So what we do is that we extract the RNA from here. We just try to take all the blue and red RNA. So the blue RNA and the red RNA, which will be more. To be honest, I don't know how exactly they do that. But what's happening is that we convert this to cDNA. So again, we will have DNA now. So this was RNA here, but we have DNA now, which again will be a few blue RNA and a lot of red RNA. So this is of course a simplified image of what's happening. We have alternative splicing and so on, but for the moment let's forget all these details about alternative splicing and just let's focus on the fact that we have mature RNA, which means that each RNA molecule is in fact the following. If this is the DNA and here is the red gene, and the red gene comprises a few exons, so exon number 1 and exon number 2 and exon number 3, it is possible that the mature RNA will take, for example, exon 1 and exon 2, and will neglect exon 2 here, exon 1 and exon 3. So it will be this thing, exon 1 and exon 3. Just as an example. And this molecule here is this red molecule that I have around here. Okay? And the similar thing is true for the blue one. Okay, now this mature thing, this mature RNA, which is DNA in fact, so this mature cDNA that came from an mRNA will be sonicated or will be split if it is too large in smaller pieces, so in fragments again. We have talked about fragments, I will not say much about that. So by fragments I mean that if this is a large one, then maybe it will break in some pieces, right? 3 pieces, for example, if this is very big. And in some cell it might break like that, in another cell it might break like that, and a longer here piece, and so on and so forth. So from different cells we have different breakages from the same cDNA molecule. Okay, and these fragments will be sent to the sequencer. And in the sequencer we will have what we already know, which means that if this is a, so if we take this fragment so big, then we will sequence it from here, if it is single end, or from here and here if it is paired end. Okay, and we will do that for all fragments and for all these cDNA molecules here and here. Alright, now imagine the following. I will draw this again. We have all the cDNA molecules here. These are, let's say that these are fragments now. The red one, and we have a fewer blue, less blue actually. So these are again fragments, right? Fragments, all of them are fragments. And this will be sent to the sequencer. So this is the sequencer. And we will get reads. Now what will be true for the reads is the following, that since we will try to sequence each one of this molecule here and each one of this molecule, eventually we will produce many more reads from here than from here. So again we will produce reads from these fragments, and if we see them here in this Eppendorf or whatever, we will have these reads from the blue and many more reads from the red one, just because the expression level is higher. Okay, and then we will try to map them. So I will speak about mapping because it's a special process in RNASeq in a while, but for the moment I just want to show the logic behind using sequencing for measuring the RNASeq levels. So if this is a red gene and if this is a blue gene, many more reads will be mapped here. In fact, not like that, but on the exons of the red gene, and much fewer reads will be mapped to the blue gene, just because I have fewer blue guys than red guys. Okay, now just measuring then the number of counts that I have here and here, let's say that I have 1000 reads here and just 100 reads here. And let's suppose for the moment that we have 2 exons here of the same length and 2 exons here of the same length. So all these 4 exons are of the same length. This is important actually, you will see that later. We can figure out that this gene is expressed at higher levels than this gene. To give another example, which will make things a bit clearer, let's assume that we have 2 conditions. So condition A, let's say an untreated individual so no drugs, nothing here, and condition B, treated individual, which means that perhaps we administer some drug A to this one. And we want to study if the expression level of genes is affected by the drug. Okay, let's focus on just one gene for the moment, and it will be a similar process if we have more genes. So let's focus on gene blue. Blue gene here and the blue gene here. These of course are the same genes, they have the same number of exons, they have the same number of exons here, and let's suppose that we have high expression levels here. So again, with all the process that we described before, we will map many reads here, mainly on the exons actually, like that, and fewer genes here, and fewer reads here. So it will be like that, just because the expression level of this guy is lower. So it will produce less mRNA, which means less cDNA, so less material to sequence. We will have more reads here and less reads here. So here we don't have to worry if this gene is longer than this one, just because they are exactly the same genes. Therefore, if we compare now the number of reads, and we have 1000 here and just 100 here, we can conclude that the expression level of this gene is higher than the expression level of this gene. There is a small detail that actually I omitted, is we have to take care of the whole library, so of the total number of reads that we have here, and we have here, I mean for the remaining genes. So here we have another gene and another gene and 1000 genes here, and another gene and another gene and 1000 genes here. Now we must take care of the fact that perhaps we initiated this whole sequencing thing with different amount of material of initial mRNA, just because, for example, we put a different amount of reagents or different amount of initial RNA. So we should take care of this fact, and this process is called normalization, but we will see that later on. In this short video, I just want to describe that it is possible and how it's possible, what is the logic behind using sequencing to obtain insights into the expression level, the transcription level of mature mRNA of the cells. In the next video, we'll discuss normalization. We will discuss the problem of mapping things only to exons. So the mapper, the aligner, should be splice-aware aligner. And then in another video, we will discuss how we quantify the different transcripts that we might have. These will be all in other videos. Thank you very much.